Welcome here, everybody. I'm going to start here in a couple minutes. Hope you're doing well. Hey, everybody. We're going to start here in just a couple minutes. Thanks for tuning in today. I'm going to give it a little bit of time for people to join on here. $12:30 on the dot Pacific time, and I hope you're doing well out there wherever you're tuning in from. I know the weather situation out there can be a little nefarious with the winter storm warnings that I've been hearing about. I am not particularly influenced by this because I live in California. But welcome, everybody. Glad that you're here today. Let's go ahead and jump right into this as we have an enormous amount of material to cover. I'd like to really personally thank everybody for being in attendance today. And I'd like to say that RCCX is my baby. I've been researching this for quite some time now, and this is something that I'm very passionate about presenting on and researching and looking to a very complex problem that I'm looking to solve. So let's go ahead and jump right into this. My name is Michael McEvoy. I'm the founder of MetabolicHealing.com, and many of you know me from that role. I'm the founder of Metabolic Healing. I consider myself to be a clinician, an educator, and somebody who's working in the field in complex illness and helping to develop theories as well as therapies in the realm of navigating complex illness. Certainly in this day and age, in the 21st century, the prevalence of chronic disease is now very prevalent. And so we want to have better models to understand what it is that we're seeing. And this is one of the kind of the reasons I got into this research regarding RCCX. So my latest contributions to this chronic disease puzzle, one of these involves this RCCX theory or RCCX research, as I believe it's more research than it is a theory. I believe that there's a large subset of patients in the chronic disease population, and I estimate anywhere from 25 to 40%, maybe more, that have inherited an RCCX genotype that is contributing to their complex illness in a significant way. In our practice in my company, we're beginning to conceptualize and identify some of the lab testing markers that are useful in evaluating RCCX. And we've also developed the first wave of therapeutics for what we are calling an RCCX genotype, and looking at what some of these approaches are and what's been working and what isn't. So I'm going to be presenting a case study here on that a little bit. I believe that medical and scientific research will eventually recognize the RCCX gene cluster located within the HLA3 region on chromosome 6 as one of the most significant regions within the human genome involved in chronic disease, as we learn about the complexities of the region, as well as the genes that comprise the region and their effects throughout the physiology, we start to see a lot of overlap. I want everybody to stick around to the end because we are going to be doing a question and answer, and I'm happy to field your questions. You can always pop questions into the chat, and we'll be jumping on there. So RCCX. First of all, the RCCX letters represent the first letter of each of the genes in the region. So the R is the RP1 gene, the C is the complement C4, the next C to that is the CYP21A2, and the X is the TNXB. They call this thing RCCX. And this is a graphic representation from, I believe, one of the type 1 diabetes study that was looking at the association between low RCCX copy number and type 1 diabetes susceptibility. What we need to understand is that over recent years, there's been an explosion of research into human genome, as we all know. And we are still learning about genetics. Our understanding of DNA is a rapidly and continuously evolving field of which we are only at the very beginning, in my opinion. And there are certain hotspots within the human genome that are very, very significant because they are known to contribute to genetic susceptibilities to diseases, to complex illness. And the old models of looking at disease are really less relevant than they are today. You know, 1 + 1 = 2, pathogen equals disease, single virus, single vector, single disease. Those models are no longer relevant because complex illness involves a convergence of thousands of variables simultaneously. Now, we know that genetics plays a significant role in disease susceptibility, but there has been many attempts to try to isolate singular nucleotide polymorphisms to associate with different illnesses. And while there's been an enormous amount of research into that, there's overall been not a totally successful attempt at being able to figure out how to modulate genetics or even to how to utilize genetic data in a meaningful way. When we talk about the RCCX cluster, we are not talking about the normal rules of genetics. There's a lot of things that are occurring in this region that are very, very complex and very unusual. And they do exist in other parts of the genome, but the fact that all of these anomalous things that are taking place in the RCCX cluster in a single cluster of 4 genes is really unusual. So RCCX is what's known as a copy number variation, and copy number variation gene sequences comprise about 4 to 9% of our DNA. So they're not that common, but they have a huge impact on susceptibility to diseases. We know other copy number variations in the genome, like the ones associated with Huntington's disease and Fragile X syndrome, can lead to huge genomic instabilities. So the RCCX region is going to comprise a series of pseudogenes, miniature genes that are not active but can affect transcription and protein translation. We have in here what are known as the chimeric pseudogenes. And what's interesting is that the 4 genes that make up the RCCX cluster, they tend to behave like one gene as opposed to 4 separate genes. They are, if you start to look at the research, the genes, they can share domains in genomic regions. They can overlap in function. So for example, the 35th intron of the C4b gene contains the transcriptional regulation for CYP21A2, the gene that makes cortisol. So one gene can control another gene, and the regions can overlap. These are really anomalous things, but they occur within the RCCX cluster. There's a HERV-K retrovirus located within the C4 gene, and some of the recent research on schizophrenia has shown that the HERV-K retrovirus can affect how the C4 gene expresses itself as a protein in the brain and nervous system, which is leading us to a better understanding of how in some of the mechanisms that are going wrong. And then the 4th gene in the cluster, known as RP1 or STK19, contains a retrotransposon, which is a retroviral-like element. So these are a few of the anomalous things that take place in the RCCX cluster. I have created a practitioner training course, which I'm going to explain a little bit more later about. And it's going to, basically, I created this course out of the necessity to really bring light of this RCCX situation to the clinical awareness. So the TNXB gene is your extracellular matrix glycoprotein. It is one of a series of glycoproteins in the ECM that regulates the anti and pro and anti-adhesion of the connective tissue. It neighbors the CYP21A2 gene on the cluster, and this is the gene that produces cortisol and aldosterone. So of all the endless debate about the effect of cortisol in chronic disease, from both sides of the aisle, whether you're alternative practitioner or conventional medical practitioner who believes that there's no such thing as adrenal fatigue, whatever the case may be, the fact of the matter is that the gene that produces cortisol exists in one of the most complex regions of the human genome and is highly unstable for a number of reasons. The gene also produces aldosterone, so it's going to regulate salt balance as well. CYP21A2, the gene to the left of that, and the neighbor is complement C4, which is a critical part of the innate immune system. But the more recent research has identified that C4 is critical for synaptic and dendritic pruning in the nervous system, and this is critical as it's related to schizophrenia, to autism, and to bipolar disorder. And then, as we've mentioned, there's a retrovirus within the C4 gene that actually can regulate the function of it. And then the fourth gene, we don't know what it does yet. It's a threonine kinase, but we don't exactly have any knowledge of what RP1 does yet. So identifying the RCCX phenotype and their families. This is the URL to an article that I authored that was published just a month ago. A very dense, very thick article, but one that I'm trying to get out there into the clinical world as much as I can, because it illustrates the major problems, the major complications, the major remarkable things that are occurring, as well as how we as clinicians can find the patient in the population and their families who have this going on. So that's a very useful article that I've put out there for the public to read. It's, again, designed to really, really research-oriented more than anything. This is a list of what I am looking for when I am trying to identify the RCCX phenotype. And what I want to start by saying is that in order to find these people, it's equally as important to identify the traits, not only in the patient who suffers, but the entire family. So one of the things with RCCX is it contains this what's known as unequal crossing over. An unequal crossing over can significantly influence familial inheritance, where you will normally see people that have very similar disease traits all in the same family, even within cousins, can segregate in that way. And so we can break down a lot of the symptoms based on the function and or loss of function with each of these four genes. The tenascin X being the extracellular matrix link. The first thing that you want to do clinically is, does the patient exhibit signs of joint hypermobility syndromes, such as Ehlers-Danlos syndrome, joint hypermobility, hyperextensibility, skin hyperelasticity, which is another major tipping point that TNXB may be involved. Now, I want to start out by saying that just because somebody does have joint hypermobility does not necessarily mean that they have anything going on with RCCX. I've identified many people that have diagnosed EDS, hypermobility, but they don't fit the category. The family doesn't seem to have the characteristics, and none of the other evidence exists. So just because you're hypermobile doesn't necessarily mean that, but it is one significant indicator for RCCX if you do. I then move on to the next question. Is there anybody in the family, yourself included, that has cardiac valve-related problems, esophageal valve-related problems, because the tenascins can play an important role in valve-related functions, and then organ prolapse, uterine prolapse, intestinal prolapse. There's one other one in here that I didn't mention, is vesicouteral reflex, which is basically retrograde urine flow. That's a huge tip-off of tenascin X. The second part is we're going to look at the functions of what can go wrong with 21-hydroxylase, CYP21A2. Now, the scientific literature has not really done, there's not a ton of research on CYP21A2 other than studying congenital adrenal hyperplasia, which is still considered a quote-unquote rare disease. But this is directly linked to mutations of CYP21A2. Essentially, what happens in that condition is that an individual does not produce enough cortisol. And so because of this, the 17-hydroxyprogesterone gets shunted to form androgens. And so this can lead to ovarian cysts, PCOS, hyperandrogenism, and all of the symptoms associated with that, facial hair, acne, all of those hyperandrogenic symptoms in a female, for example. But CAH is really the only main condition studied with 21-hydroxylase. That's really ridiculous because we know from studies going back to the 80s that mild deficiencies of CYP21A2 have been observed in European populations. So we know 21-hydroxylase is something that it should just be studied more in relation to other conditions. But there just hasn't been enough research into that gene yet. But I believe that as they start to do that research, they're going to find some huge hits because we know that low cortisol is a big problem. And by the way, I should say that most people with CYP21A2 mutations will tend to produce a phenotype of low cortisol. However, even with CAH, there have been cases where high cortisol have been observed, and that's published. So it's a very anomalous gene that we need more data on it. We know that it can actually express in the brain as part of a neurosteroid pathway. So some of the symptoms that may be associated with 21-hydroxylase, problems, poor stress tolerance, salt cravings, ovarian cysts, endometriosis, high or low cortisol, high or low high androgens or estrogens. Autoimmune disease, this is a huge, huge component of helping to identify the RCCX phenotype and in their families. So this list of these particular autoimmune diseases are all linked to complement C4 in one way or another. Lupus is probably the top of that list. 75% of lupus patients show a C4 deficiency. 48% of lupus patients show joint hypermobility. But nobody's actually looked at the association between RCCX phenotype and how all of those fit together with a lupus patient. I'm going to share with you some biochemistry later when we're going to show the potential effects of C4 deficiency and the effect that that's going to have on the coagulation cascades as well as B cell and T cell signaling. Type 1 diabetes, celiac, Crohn's, juvenile dermatomyositis, Graves' disease, RA, myasthenia gravis, ankylosing spondylitis. Look for these diseases in the family. If the shoe fits, it probably suggests RCCX involvement. So if they have hypermobility and or TNXB symptoms, they may have CYP21A2 symptoms, and either they or their families have these autoimmune diseases. What you're going to normally see as you analyze the families is that these particular autoimmune diseases, they run in parallel in families. Mother has lupus. We're going to look at the case study later, which is just obvious. The other significant component here is that the C4 gene is also expressed as a protein in the brain and nervous system. The one study that was done here, I believe it was an NIH study, found that the C4 protein was the most significant protein that regulated the relationship between neurons, controlling synaptic and dendritic pruning. So again, it's common to see the patients that have autoimmune disease in the family will also have individuals in the family with schizophrenia and bipolar disorder. The link there is aberrant C4 expression in different parts of the body. And I'm postulating that's because of an RCCX phenotype. Autism spectrum, that's another one. 2 separate studies will look at that, C4b null alleles with that phenotype. Let's break it down. So again, TNXB, extracellular matrix glycoprotein. There's been a recent study on the association with tenascin X and schizophrenia. So that glycoprotein can express in different tissues. The protein itself is found in the gut mucosa, the skin, the muscles, the joints, the adrenal glands. And so while the haploinsufficiency of tenascin X is uncommon, we believe that TNXB adverse events of different kinds can cause problems. You don't have to be haploinsufficient. You can still have TNXB problems regardless. The other deeper things we need to appreciate is that as a protein, tenascin X regulates the bioavailability of 2 important growth factors, transforming growth factor beta and vascular endothelial growth factor beta. So these are very, very critical. So TGF beta, this is one of the most ubiquitous cytokines. but is a huge master regulatory cytokine in the immune system, also participates in stem cell development and embryological development. So that begs the question that what could go wrong in a person that has tenascin X deficiency or problems in TNXB. This could have a direct effect on immune function, on stem cell function, on embryological development. Remember that in hypermobility syndromes, it's common to see abnormal brain development. Hypertrophic amygdala has been observed, has been published in EDS, in JHS. That could be traceable back to aberrant TGF-beta signaling. Those studies have been done in mice where they found that the enlarged midbrain was the consequence of a loss of TGF-beta receptors in the brain. So we know that these things could be linked. And we also know that TGF-beta plays an important role in the synaptic pruning process and actually works with C4 and C1q, the complement in the pruning in the brain. So there's deeper associations here between what could possibly go wrong when TNXB is mutated or is not expressing appropriately. CYP21A2, salt wasting, poor stress tolerance, ovarian cysts, common female presentation. She's hypermobile. She feels like she's craving salt. She has low blood pressure. She has a terrible intolerance to stress. She has PCOS ovarian cysts. That is and then you look at the family history of the autoimmunity, textbook RCCX phenotype. We see that kind of a person a lot. 21-hydroxylase is expressed in the adrenal glands, but it's also found, the RNA of it is found in the brain and nervous system. Postulated that it could be part of the pregnane neurosteroid pathway. We don't exactly know yet. The transcription of 21-hydroxylase, the DNA to RNA conversion, the start points of that exist in the neighboring C4b gene. So if C4b is affected in some adverse way, that could directly affect cortisol. I believe that's occurring in lupus. There's a lot of cases where we see low 24-hour urinary cortisol and low total C4. So as I mentioned, complement C4, key part of the innate immune system. The complement cascades depend on C4. We know that the low RCCX copy number is associated with low C4. And we're going to show a recent paper later that shows how C4 expression is regulated by the Tregs and the TGF-beta, and how this trifecta is working together. And again, C4 is strongly linked to the pruning process in the brain and nervous system. So C4 deficiency has actually been described as one of the most common immune deficiencies worldwide. We have to look at the complement immune system when we're talking about complex illness, because this is the complement immune system, part of the innate immune system, is one of the oldest immune systems of our human body. Throughout the evolutionary process, it's believed that actually the lectin pathway, which you're seeing here in the middle here, which involves mannose binding lectin and phycolin, which is a collagen-like compound. These pre-proteins are circulating throughout the blood, and they're derived mainly from the liver, produces these complement pre-proteins. And they're basically looking to grab onto bugs and viruses and other pathogens. And that creates the complement cascades, where you start to break apart the bigger proteins, and they start to split off into smaller parts. And this eventually leads to the signaling of B cells and T cells, right? And so as you can see from the illustration here, if you could see from the illustration here, the C4 and the C2, which you see in the blue, and C2, mind you, is just a couple doors down from C4 on the HLA region in the RCCX cluster. So it's also in the HLA3 region. But C4 and C2 are necessary for C3 in the C3 convertases. This then splits off into C3b, and then the subsequent formation of C5, 6, 7, 8, and 9 all are involved in basically creating this quote-unquote membrane attack complex for cell lysis. So think of it higher up the chain. What happens if you don't have enough C4 present, such as in a lupus case, which is so strongly linked to C4 deficiency? The ability to form this lysis and membrane attack complex is going to be impaired. So we have to think about it in this context. This is some biochemistry for you. And again, this is from Keg. This is a really, really good way of looking at this. And you can study this stuff all day long. But the C3 convertases are what form from the C4 and the C2. And I highlighted some of the important elements in pink. We're looking at the coagulation cascade to the left and the complement cascade. Well, I've always wondered the relationship between why is abnormal blood clotting seen when there is innate immune system problems. And one of the links is thrombin. As you actually follow the arrow on the left here, thrombin can participate in the formation of C5. But again, I want you to think in terms of what happens if there's not enough C2 or C4. How is that going to affect the cascade downstream? So that could potentially implicate an upregulation of thrombin because thrombin can contribute to the formation of C5 and C5 activities. So in that case, you may see more thrombin synthesized as a result of that. As a result of the complement cascades, when C5 is split off, so again, you need the complements to be all working in concert. If you've got something deficient upstream, that's going to push and pull things downstream in an adverse way. But this all can lead to B cell signaling mechanisms. And this is the next page, basically. So this is CD19 and CD21, which are these are B cell. These are on the surface of B cells that are going to activate your antibody system, right? And so here's the pathways for your B cells. So again, if the complement cascade is aberrant, and again, I'm saying in the case of when we have an RCCX phenotype, if there is not enough C4 being produced, this could adversely affect the ability of B cells to work right appropriately. But there's actually more than this. As we go to this next study here, this was a study done with they took the blood from different patients, including with lupus patients, and they wanted to look at what would happen when they incubate the cells and what the relationships are between things. And basically what they found was that there's a direct relationship between complement C4, T regulatory cells, and TGF-beta concentrations. And the significance of that, as it relates to the C4, is that if you have low C4, your ability to have utilization of your TGF-beta, which again, I want to point out, TGF-beta is an anti-inflammatory cytokine. It's the most immunosuppressive cytokine. Even though it is involved in pro-inflammatory activities, it's the most anti-inflammatory. And it works to activate the Tregs and the FOXP3 transcription factors. If you have somebody that's predisposed to autoimmunity, you absolutely need to be thinking about how to modulate their Tregs. Tregs are the types of soldiers, if you will, that will basically come in and say, ceasefire, ceasefire, ceasefire. We're calling off the shots. We're going home, turning off the inflammation. That's what your Tregs do in the context of autoimmunity. They're critical for self-tolerance. They're critical for oral tolerance, to be able to tolerate bugs in the gut mucosa. The Tregs, TGF-beta, FOXP3 system are critical for preventing hyperinflammatory and autoimmune activities. So there's a wide list of implications of what can go wrong if there's not enough C4 that gets produced. And it can set the stage for autoimmunity very easily. This is a significant study that was looking at the C4b, there are actually two studies, I should say, looking at C4b null alleles and autism. And what you really need to understand here is that these are two good studies that were looking at around the same number of subjects of autistic patients, taking from two different geographic locations, one from Egypt and the other in the United States, and that they found very, very similar findings, was that around 40%, so the first study showed 42.4%, and the second 37.5%. They're looking at autistic kids and how many of them had the C4b null alleles. So on the RCCX cluster, the complement C4 gene is divided into two isoforms, C4a and C4b. And the expression of these proteins will vary from tissue to tissue. But what we know is that in autism, there are problems in the synaptic pruning process. Autism tends to feature increase in excess of synapses versus schizophrenia, which is an excessive amount of pruning of synapses. So the synaptic pruning process can be likened to the pruning of a fruit tree that is overgrown. We have these mechanisms in the brain that trim the synapses back. You're getting a synaptic haircut, if you will. And the complement proteins play a critical role in this synaptic pruning process. Well, the C4 protein has been identified in at least one study as the most significant of the complement proteins. C1q and C3 are also involved. And TGF-beta is involved as a cytokine involved in the signaling of it. So again, think about these things in terms of the relationships. So what these studies both found was that nearly around 40% of, if you average the two together, around 40% of autistic kids carried the C4b null alleles. And that's a pretty significant association. In the second study here in red, the frequency of autoimmune diseases in families of autistic children was 40% was significantly higher than the healthy controls. Well, what they didn't actually study was the actual relationship between the C4 gene in these autoimmune patients of the family kids, the kids with autism, because it would have tied directly back to RCCX. Because C4 is not only critically involved in the innate immune system, but it's also involved in the synaptic pruning process. So this is really significant. The other thing is that this genotype, known as the C4BQO, is they identified a huge risk factor for autism. That this genotype tends to produce higher cortisol levels, higher HPA axis activities. So it's one particular RCCX genotype that we need to be looking for. I want to say that there's really no way yet to get your RCCX cluster genotyped. It's not something you have commercially done yet. I'm working on helping to move that forward. That's one of the side projects I'm involved in doing, is trying to get a lab. Let's do the sequence and let's do the deeper sequence of reads so that we can actually prevent these diseases from taking place. If we had a way to predict predict a disease, we could prevent it. And if we thought about it like that, that means that all of these environmental triggers, and I'm going to share in the case study, I'm going to show you why I think the environmental triggers, environmental toxins and pathogens, is just too much for people with these undesirable RCCX genotypes. It's too much. The immune system can't take it all on. They're not programmed to do it. And the level of toxicity that we're all being exposed to is so great that the stronger will survive, unfortunately. And so, but if we had a way to predict who would be more likely and more susceptible to developing autism, and to show associations between how various toxins and heavy metals and pathogens may be significantly contributing to a pathology, that's really, really important to me. And so, to me, the research that we need to conduct going forward is so critical. Here is a recent paper published in 2016 on schizophrenia and the complement C4 gene. And again, remember that C4 modulates the synaptic pruning process, which is why it's so strongly linked to autism. But here's the schizophrenia article. And what they found was that the number of C4a and C4b copies is strongly linked to schizophrenia risk. Now, you've heard a lot about retroviruses lately, and Dr. Klinghardt and Judy Mikovits have been recently really putting out a lot of information about the involvement of different retroviruses. And So, you have exogenous retroviruses and you have endogenous retroviruses. The endogenous retroviruses, they have evolved as part of our DNA. Somewhere like 8 to 13% of our genome are comprised of human endogenous retroviruses known as HERVs. And you've got different HERVs, and they do different things. They actually play an important role in how the body works. For example, pregnancy, in order for the placenta to attach and to prevent the immune system from attacking it, retroviruses can prevent that from happening, can inhibit the immune response. That may be partly how retroviruses are associated with cancer. Similar mechanisms of how immunosuppression can occur in cancers through retroviruses. And there's a lot of different diseases that have shown to have the RNA expression of different endogenous retroviruses. Autism, schizophrenia, multiple kinds of cancer, autoimmune diseases have all shown to have the RNA of endogenous retroviruses that mobilize from our genome and somehow end up in our blood. Dr. Navio, in his cell danger response research, has pointed out that in the 6th phase of his cell danger response, there are the mobilization of endogenous retrovirus and retroviral elements from the DNA that can get mobilized. And I've looked at, well, what are they doing? They can stimulate the innate immune system. They can do different things with immunological signaling. So the fact that the C4 gene itself has evolved to contain the HERV-K retrovirus in it is very, very significant. And it appears from this study here that the presence or absence of the retrovirus will affect the function of the C4 protein and the expression of it in different tissues. So, and that could obviously play out in a good or bad situation. I think that what this paper is showing is that there is an association between HERV-K activity and the function of C4A and C4B in the pruning process of the nervous system. So very, very significant. All right, I want to take you through a case study here. So again, I want everybody to come away from this call understanding who to be looking for with the genotype. Because again, we don't have the ability yet to do the genotyping on RCCX. What we can do is we can look at associations in families and the symptoms of the patient. We can also be looking at a couple of lab markers right now, and I'm hoping to expand that list as we go forward. But I want everybody to come away with a better understanding of how to isolate and identify these people in the population. If you signed up for this webinar today, you have the opportunity to actually look at that survey that we sent you. And that survey is what we use as an initial screening to identify an RCCX phenotype. And again, it's all based on the categories of the functions attributable and even the conditions and diseases attributable to each of those RCCX-related genes. So this case study is going to give us an understanding of how the genetic susceptibility can link together with the environmental exposure to create a chronic illness kind of a situation, milieu, if you will. Just to address a single question that I want to be clear about what does the RCCX copy number mean? So as a copy number variation, the number of copies that exist, a person is genotyped in the scientific research on RC, because they can genotype you if you're in a study, but not in the clinic. But there's 4 ways of defining that. You're either a monomodular, a bimodular, a trimodular, or quadrimodular. And the number of C4 copies that you have, duplications of the C4 gene, defines whether you're monomodular, bi, tri, or quadrimodular, your RCCX genotype. And what's been observed is that the lower the copy number, if you have a monomodular RCCX genotype, there's a much higher risk for autoimmunity. And that lower copy number tends to correlate with the low C4 protein levels. Now, just to add more complication to it, because this is not simple stuff, that the size of the C4a and C4b gene can also influence the protein level. So you can have a C4a short, C4b long, C4b long, C4a short. And so the size can affect it. And the size is also going to affect the actual HERV-K retroviral. If you're short on one of those, the retrovirus could be low. And so the protein expression can be changed. So these are complicated things that way too much for this webinar today. But just know that low RCCX copy number is associated with autoimmunity. And that means that you've got fewer copies of the C4 gene. So this is a 30-year-old female patient recently diagnosed with lupus. She had extreme pain in her extremities, and she was having a difficulty just getting dressed. Curiously, she had a persistent cough. They were living in a rental unit where there was a water pipe that was recently ruptured, which is suspicious for mold. We know that mold is involved in immunological signaling. We know that a lot of the work of Dr. Shoemaker and those that have followed in his footsteps have looked at C4a and C3a, which are split proteins of the C4 protein, in relationship to mold, as well as Lyme. So we know that the innate immune system is involved in clearing and removing pathogens and toxins like mold. Here's the patient's family history. I want to say that this was a case study that I was collaborating with her functional medicine physician, and I was educating her physician on RCCX and feeding her that information as we were helping to create and construct protocols. So her aunt had lupus. Her uncle had myasthenia gravis and type 1 diabetes, both strong links to RCCX. Her cousin had type 1 diabetes, has strong RCCX association. Her grandmother and great-grandmother both had bipolar diagnosis. That's a suspected RCCX association. There's not enough data and research on bipolar disorder and complement proteins. There needs to be, because you're going to find associations. That's hopefully future research to be done. Mother has Crohn's and Hashimoto's. Hashimoto's not studied, but Crohn's definitely is associated with C4 and RCCX. Uncle with myasthenia gravis. That's definitely a low C4 RCCX associated disease. So you can see the associations with the mental illness as well as, I should say, psychiatric presentation and autoimmunity. Here's her stool test, just to point out highlight some of the potential problems. This is the GI map, which is a good stool test because it's a PCR. I like it. So you can see the patterns for dysbiosis, the potential autoimmune triggers like the Prevotella. Now, I want to again point out the significance of gut bacteria. Now, we obviously know that they play a significant role in so much of the immune system and behavior and neurotransmitters and all that. But what we need to appreciate is that what can quell and prevent a lot of the hyperinflammatory responses in the gut mucosa to these bugs are the T regulatory cells. If you do the research on the Tregs and FOXP3 transcription, they are so significantly involved in what's called oral tolerance factors. In other words, preventing these bugs from creating pathogenic hyperinflammatory responses. You can have many people with parasites, many people with all kinds of different so-called pathogenic microbes in the gut, completely asymptomatic. Now, it's because you've got things that prevent that hyperinflammatory response, like the oral tolerance factors, FOXP3 Tregs. Now, low C4 is going to have a direct effect on TGF-beta and in this signaling. So we know that if you're seeing somebody with autoimmunity and they are suspected RCCX, you have to think about the Tregs being downregulated. Again, remember that study that showed the low C4 is associated with low Tregs, and we need Tregs to calm the immune system. Here's the blood work. Here's the low C4. Now this lab, the LabCorp, if this is not LabCorp, but LabCorp's range for total C4s, I believe, the bottom of it is 16. She's in single digits at 7. And so the C3 you also see is on the lower side. Remember that you need C3, you need C4 in order to synthesize C3. 75% of lupus patients show C4 deficiency. And here's the antibodies. and then the B1 was also low. So these are significant findings from her tests. Here's the Dutch test looking at the 24-hour cortisol. You can see a very, very low 24-hour free cortisol, dangerously low. Now, the CYP21A2 gene within the RCCX cluster is the gene that converts 17-hydroxyprogesterone into cortisol. And if there is a defect in that gene, or even in the C4b gene, in the 35th intron of the C4b gene, because that gene, that region of the C4b gene controls how you convert the DNA to RNA of 21-hydroxylase. And again, that's just to highlight, this is just to highlight that relationship. Intron 35 of the C4b gene controls the transcription of CYP21A2. That's an unusual phenomenon to happen, but that's occurring within this RCCX cluster. So we suspect that, again, low C4 and low cortisol, there's a direct relationship there that I'm saying is related to the RCCX overlapping shared domain situation that's occurring within the region. Urinary mycotoxin test from Great Plains. So we see the ochratoxin is elevated. That's penicillium or aspergillus, which are products of mold. So environmental trigger combines with the genetic susceptibility, and you have the inability to generate self-tolerance. You have a hyperinflammatory response. Here's the other one. So just to summarize here, this hypothesis, probably a low RCCX copy number, and probably the monomodular genotype that we know runs with lupus. She is C4 deficient. That lines up with our theory. She is very low in CYP21A2 activity, given that the cortisol is low. So we suspect it's because of this 21-hydroxylase in the 35th intron scenario. We suspect that her Tregs are low. Her ability to differentiate the Tregs from the dendritic cells is reduced, is impaired, because she doesn't make enough C4. And there's that association. Furthermore, her TGF-beta level might be dysregulated in the tissue, because as that study also showed, that the TGF-beta concentrations also tended to be affected negatively. TGF-beta is immunosuppressive as well, and it also is what's activating the Tregs. The mold is the epigenetic trigger that it causes the RCCX weakness to express in her case. So this was the protocol that was constructed for the client. Her diet, 95% gluten-free, dairy-free, lower lectin. I found that the lower lectin diet with RCCX is potentially very useful. So remember that the C4, the innate immune proteins, they're binding lectins. Mannose binding lectin, MBL, is what's on the surface of those proteins, and they're looking to grab on to lectins. And so the lower lectin diet could potentially be of value here if you're working with somebody that's low C4 and or RCCX. One of the big takeaways that I found useful, and this is one case of several now that we've observed, is I have to give kudos to the Microbiome Labs gut protocol products, because they've shown to be very useful. There's a number of probiotics that can regulate Tregs. Bacillus subtilis is one of them. Lactobacillus reuteri is another. Bovine serum immunoglobulins are a third. So in this case, this gut protocol was very significant. So the megaspore, the HU58 and the megaspore. So the HU58 is the Bacillus subtilis, which is the Tregs promoter, and it also balances the Th1, Th2 systems. Megaspore contains Bacillus subtilis in it. So there's a lot of Bacillus subtilis here. And she's also taking the megamucosa, the mega-IgG. Those are immunoglobulins. Those can also regulate the T regulatory cells in the gut lining, in the gut mucosa. Prebiotic, digestive enzymes, and then she was intermittently taking the GI detox binder from biobotanical research, again, to assist with the mold removal. Those are in the mycotoxin excretion. A number of different antioxidants were added here, including hydrogen tablets, Avmacol, which is a sulforaphane promoter, helping to switch on that NRF2 pathway. She was doing a little bit of liposomal glutathione without rocking the boat. She tolerated it well. And as you can see, some of these other antioxidants here were potentially useful. I added in for cortisol support vitamin B5 and choleosphorxolin. Choleosphorxolin is a cyclic AMP promoter. Cyclic AMP is one of the cofactors for CYP21A2. And vitamin B5 is probably an indirect cofactor as well. I'll look at the more detailed protocols when we review that. This is her protocol. Minerals and electrolytes are really important for RCCX phenotypes because, first of all, if you're low in cortisol because of 21-hydroxylase defect or adverse event affecting low cortisol, there's a higher probability you're losing your sodium because aldosterone synthesis is going to likely be impaired. So you need to get the salt back into the body. You have to regulate the charge of the tissues. The extracellular matrix is impaired for any number of reasons. Lupus and RA are rheumatic diseases, connective tissue diseases. If hypermobility is on deck, one of the most important things you need to be looking at is the electrolyte balance in the tissues. And sodium combined with hyaluronic acid in the connective tissue is what brings water to the tissue. It's what hydrates and bathes the tissue. So that was a key component. You see some B vitamins here, including the benfotiamine, which is the B1. And the LDN was very helpful for her to help manage the pain. These other therapies were also very significant for her. Cryotherapy, which she reported significantly helped to calm the major inflammation. Hypnotherapy, trauma release therapy were used, also considered to be very useful in her case. Doing some sauna therapy and some very important lifestyle stuff, blocking the blue light, early morning sun exposure, no Wi-Fi in the home. And that, over the course of several months, 95% improvement with this particular case. So a really, really positive thing here. Again, I want to just summarize that if you're working in autoimmunity and you suspect RCCX and you're seeing, and I want everyone to run the total C4 if you're suspecting it. Total C4, if that's coming back on the low side or low, we have to think about the effect that that's having downstream on B cells and on T cells, especially the Tregs and the TGF-beta. What if the TGF-beta is high? If it's high, it doesn't necessarily mean that it's truly high. How do I clarify that? TGF-beta is differentiated in the connective tissue. The blood is the expression of it in the blood. You can have high TGF-beta in the blood, but the ability to utilize it in the tissue is impaired, or to utilize it at the cell level is impaired. We can't get that sensitive data by looking at a high or low blood level of TGF-beta. Instead, what I've proposed in my article is what we need to be looking at is the relationship between TGF-beta, CD24, CD25, which is the Tregs, we need to be looking at those markers together in combination. Because there's an intrinsic relationship there between those. That will give you more data than just looking at the TGF-beta by itself. So I want to review protocols. I want everybody to take some notes here. This is some good takeaway information. So the number one thing you need to know is that if there's environmental triggers on board, you've got to address that. First, investigate as a matter of priority 1 through 4, mold, Lyme, Bartonella, and virus. You've got to do it because their ability, the RCCX phenotype's ability to modulate here is impaired. How can we support tenascin X? If there's a hypermobility component, minimize the toxin and pathogen exposure, which can lead to the formation of DAMPs, the breakdown of the connective tissue. We can also use polysaccharides, aloe vera, red algae, aquamin, mushroom polysaccharides. These are my top 3. Polysaccharides are the basic food stuff of the connective tissue. This is what your ECM, proteoglycans, and collagen is comprised of at the fundamental level are amino acids and sugars in polysaccharides. We can also use, I'm experimenting with growth factors. Megacomplex GF, chicken embryo, contains oligopeptides, fibroblast growth factors. We can use things like near-infrared sauna between the 800 to 850 nanometer spectrum. CBD oil not only modulates neuroinflammatory stuff, but also fibroblast cells and can help to produce collagen. C4. First of all, if the C4 is low, I want you to regulate the Tregs because that's critical for immune tolerance. And Lactobacillus reuteri, Bacillus subtilis, short-chain fatty acids, your butyrate, propionate, as well as serum bovine immunoglobulins are the 5 I've found that will regulate your Tregs. So it's like a workaround for if the C4 is low, we modulate the Tregs to try to get the system back up and running. And so far, this, and we've got a couple other case studies where the Bacillus subtilis seem to be a very, very significant improvement with the autoimmune component. CBD oil may be useful as well because that also might modulate the Tregs. And again, you got to lower the toxic burden if the C4 is low because their innate immunity is impaired. We can use astragalus polysaccharides. If you can use astragalus, don't use tincture from alcohol. Won't work. Polysaccharides are water soluble mainly. So use either a water dilution of astragalus or powder or capsule. There was a good study that looked at elevated the astragalus increasing C4 and C3 in myasthenia gravis, which is an RCCX disease. 21-hydroxylase, how do we modulate? Lifestyle to modulate the stress response. Got to pay attention to hydration and electrolytes. There's a lot of reports that emotional processing therapies are very, very useful for suspected RCCX phenotypes. This includes EMDR and DNRS, hypnotherapy, and then even doing emotional processing therapies before doing it, using LDN actually like 20 minutes before has been shown to be useful as a way to modulate the limbic response, if you will. Stirring up emotional memories can charge the hypothalamus and the amygdala, as those are particularly rich in cortisol receptors. And if you're not making enough cortisol, or if the expression or the rhythm is off, it's a problem. Potential promoters of 21-hydroxylase. Cyclic AMP, so we use coleus. B5, salt, because angiotensin 2 can promote CYP21A2. And then insulin signaling. I don't know if ketogenic would be the right thing. There's no diet that's perfect, but we know that insulin can also stimulate cortisol production, and we could use inositol as a workaround as well. There are some things that can lower CYP21A2 because there are some phenotypes that have high cortisol. But vitamin D, there was a single study that showed that it had an inhibitory effect. The calcitriol had an inhibitory effect. We might also consider GABA, glycine, B3, and ashwagandha as a way to modulate high cortisol. But look for things that could be inhibiting cortisol if it's low, too much, if they're taking thyroid hormone, or if they have excess thyroid hormone, or growth hormone, for example. Those could be significant. So I hope that these are useful for you. So again, my name is Michael McEvoy, and I wanted to just spend the next few minutes here talking to you about my training course that I developed for clinicians like yourselves. It's my goal to unearth as much information and research about this complex gene cluster. And I believe that RCCX will be eventually recognized as one of the most significant hotspots in the nuclear genome that is directly related to the cell danger response, as well as a huge genetic susceptibility for many people suffering with chronic disease today. This was a single case study, but we can go through many others that will help to improve your understanding. So this course is a 6-hour training course that is going to actually be soon updated. I'm going to add more material to it. And I teach you how to basically identify the phenotype in the population. I'd like to poll the audience right now before we get into the Q&A. How many people right now think that they have how many patients right now have RCCX in your estimation, thinking about the phenotype? So 1 to 5, 5 or more. And so I'll pop that up there. So go ahead and throw that. There's a poll for you there. If you have any questions, you can throw them into the chat as we're going to get into some of this now. But so the RCCX course, definitely wanting to, you can go ahead and check a look at that link right there. I just put it up here. Everyone, if you get it today, it's $120 off using coupon code RCCX120. That's RCCX120. You can click the link right now to get the course. And this is a little bit of a preview from it. So you have, of course, lifetime access. There's no timelines or deadlines. my goal is to educate as many clinicians as I can about this cluster, and to do it in a way that helps them to help their patients in ways that were never before possible. Because of the fact that there are so many links between the symptoms and diseases that RCCX can cause, what we need to be looking at are the therapeutics of what is possible. And this course goes into an extensive discussion about how we can modulate the extracellular matrix and the connective tissue, and how we can modulate cortisol and C4, as well as modulating even retroviral expression, as we now have some information about what we might be able to do in order to do that, from a standpoint of helping to control the innate immune system's ability to function better. So some of the bonuses that you receive, we actually have a research forum that I've created on Facebook for all the practitioners that sign up. And we're engaging on a regular basis. I'm putting up new research. The thing with RCCX is that there's so much new findings and research that I'm coming up with that we're having to update a lot of this material on a regular basis. And so we're going to be adding an addendum soon to the course in the next few days that will help to keep your understanding moving forward. We want as many clinicians in on this as we can, because we need data. We want your feedback. We need your feedback. We need to see what's working. And so if you join our community, you're going to become part of this coordinated effort to move this research forward and to tell us what you're seeing is working in your practice when working with these phenotypes. A couple testimonials here. So what I do is I go into the complexity of the material. I present it in a way that's easier enough to understand, and something that you can apply directly in your practice starting Monday morning. So RCCX and complex illness, you're going to get a lot from it. You can go ahead and click that link. If you don't own the course yet, RCCX 120 is the receive $120 off if you use that coupon code. So we're going to let's go ahead and get into the question and answer here. If you have any specific questions, you can pop them into the chat right now, and I'm going to field as many of these as I can, as I need. So let's take a look at the polling here. How many people believe that they have an RCCX genotype patient? How many patients do they believe they have in their practice? So 3, 1, what do you think? You might need to look at this a little bit closer, because you might not have even considered this before. What I want everyone to do is to really pay attention to exactly what the family histories look like. And the more that you do that, the more you're going to start to understand who it is that's dealing with this. How long does it take to complete the course is a question somebody has. You have lifetime access. You can go through it. I would recommend going through it slowly, at least once, maybe twice. But we've had clinicians that have gone through it three times just to absorb all the material. So it's a really good course. You're going to get a lot out of it. And again, if you buy now, I'll take the $20 off the cost of this webinar today, and apply that directly to purchase of the course. Question about copper. What role does copper play in this? That is a good question worth asking. Dr. McGlathery has identified that she's found that the mitosynergy copper, which sometimes people refer to as proper copper, or copper 1, which is basically, it has a 29th electron in the outer shell, and this is supposedly the form that is used by the mitochondria in the electron transport. So supposedly that's the proper copper, but I don't know if that's totally necessary to use it or not. But I think that copper is going to push the cytochromes, and CYP21A2 is certainly one of those cytochromes. My feeling is that copper is going to have a direct effect on the catecholamines, probably more than it will on cortisol. Dopamine beta hydroxylase, for example, copper is a cofactor for that enzyme for converting dopamine to norepinephrine. And some of the symptoms that seem to be reported from copper are related to anxiety and to things like this, agitation and things like that. So copper needs to be in balance with other nutrients, including magnesium and zinc, and I would argue molybdenum as well. So I just wanted to touch back, and again, if you have any questions, pop them into the chat. There's that link. You can purchase the course right now. We're going to keep this sale going as long as we can. My team is probably not going to like that I'm extending the sale, but it'll be extended for at least, I don't know, as long as we can. No guarantees, though. It could be today. I don't know. I'd have to talk with my team about it. The question about TGF-beta is significant. So I want to go back and revisit that particularly, because what we're learning is that the fibrinogen-like domain of the tenascin X protein, the TNXP protein, which is one of the RCCX genes, the tenascin X protein has a fibrinogen-like domain, and that fibrinogen-like domain is the transcriptional regulation of TGF-beta. So if somebody does not have fully functional TNXB, their ability to properly utilize TGF-beta is going to be impaired. Now, what that looks like on a blood test is not known, because the problem is qualitative. It isn't necessarily quantitative. It isn't necessarily that they have too much or too little TGF-beta. It's more that they have a problem utilizing it and for being able to function properly. So that isn't something we necessarily can test for. The article that I recently published defining the RCCX phenotype discusses in the bottom of section of that the necessity to look at the relationship between biomarkers, not just the TGF-beta by itself. But if we look at CD4, CD25 in combination with TGF-beta 1, we'll have a much better, and C4, and those three together, that will give us a better understanding of that interconnected three-way relationship. So I hope that clarifies that particular point. And some other questions that are coming up here. One of the things I want to address with TGF-beta, with TNXB, is that the nearby CREB gene has actually been shown to control levels of TNXB. So it contains the transcriptional start points. CREB is necessary for the utilization of our cyclic AMP in our cells. And there's many associations between CREB and a number of different types of diseases, including Alzheimer's disease. Very strong link there. Question about, does hyperelastic skin always mean RCCX involvement? No, it does not. You can have an elastin deficiency that can be involved. Elastin but TNXB is known to, it's an anti-adhesive glycoprotein, and in a deficiency state, it's known to produce that hyperskin elasticity. I would look, if you see somebody that has hyperelasticity of the skin, I would look at the other RCCX symptoms and see how the whole thing lines up, right? So that's what I would say with that. Again, other questions, feel free to drop them into the chat. I've got a couple more minutes here, and we're going to wrap it up. But again, you can purchase that RCCX and complex illness course now. Save $120. You can get the course and use the coupon code RCCX120 at checkout, and you can do it. Are heavy metals a hypothesized trigger? They can be. I mean, the problem with the theory, though, is that, and the way to prove it is just simply that everyone has a lot of toxic metals. There isn't really anyone that doesn't. So aluminum is probably the most abundant metal, and everyone has aluminum and lead and mercury. Everyone has these metals in their body. You can do a urinary provocation test, but I guarantee that if you do it, everyone's going to show high metals. So now, with that said, we know that toxic elements can increase inflammasome signaling. So you have what are called on the surface of immune cells, you have toll-like receptors, and not in toll-like receptors that stimulate the what are called inflammasome complex assembly, and that process, those toll-like receptors are hyperstimulated. That leads to the stimulation of TNF alpha, interleukin 1 beta, and interleukin 18, which are basically pro-inflammatory Th1 cytokines that pathway is a trigger for autoimmunity. So one of the reasons why autoimmunity is so overexpressed in EDS and in joint hypermobility syndromes is related to the breakdown of collagen happening in excessive amount. And that excessive breakdown of collagen, basically the proteoglycans, Decker biglycan, those proteoglycans get broken apart and they stimulate the toll-like receptors on the surface of those immune cells and leading to the inflammasome assembly. So there's a huge overrepresentation of autoimmune disease in the EDS patient population. And it's, again, because the connective tissue is weak, it breaks apart really easily and you don't synthesize collagen as well as you do with other people. But again, there could be these other components like RCCX, which will, in the autoimmune phenotype, will predispose you to having low levels of C4, low levels of Tregs, low levels of TGF, low qualitative problems with TGF beta signaling. So any other questions here before we wrap it up? We got another minute or so, and then I'm just going to wind it down here. But again, great course. I'm really excited that you've done this call. I'd like to work with you more in depth, and especially on our forum, in really helping to engage this research moving forward. It really is my hope that we can take this information to the next level, and I'm working to try to develop the next level of testing, and I want to actually be more involved in the research side of it, to where we actually can bring genotyping to the market, so we can start to look at the SNPs. You can't look at your 23andMe because these SNPs don't really exist in there, and even if they did, that's only 10% of the whole RCCX picture. So we can look at familial trends, and we can look at testing, but I want to get deeper into this whole thing. So anyway, I'm going to wrap this up. Thank you, everybody, for tuning in today. RCCX and Complex Illness course is available, and go ahead and click the link. RCCX 120 is your coupon code. Go and get it. Looking forward to working again in the future with everybody here, and I enjoyed this call very much. So thanks, everybody, for tuning in. I hope it was very useful for you. Take care, everybody. Bye.